Okay, today's screencast is on the phospholipid bilayer. Now, you all should know by now that a phospholipid is this. I'm going to embed a picture, 1 second. Okay, here's our phospholipid. It's composed of 2 fatty acid tails right here and here. You'll notice one is a saturated fatty acid tail and the other is an unsaturated fatty acid tail. Now this duality, having both a saturated and an unsaturated fatty acid tail, this little kink in the molecule makes the membrane a liquid or fluid at room temperature. After we talk about saturated fatty acids in class today, for E block, this should be pretty clear to you, and B block, don't worry, we'll get to it tomorrow. So we have this hydrophobic fatty acid tail section, and then this hydrophilic head section. This phosphate group up here is very, very, very polar. So this side of the molecule loves water, this side of the molecule hates water. So all of your cell membranes are composed of these phospholipids. A cell is essentially a big ball of fat. Here's my ball of fat right here. And the environment outside of the cell is crazy. There are daggers, so to speak, of things trying to permeate the cell. There are chemical explosions happening left and right. And so inside of the cell is this nice little happy place where you can just hang out inside of a ball of fat and be protected. So that's essentially what a cell is. It's just a ball of fat serving as a barrier between this crazy outside world and this nice and stable and warm and comfortable and cushy inside world. Yes, a cell is essentially a ball of fat. Okay, so I'm going to schematicize that this phospholipid that we saw earlier over here on the next page. Okay, so here's my phospholipid. I have my 2 fatty acid tails and my phosphate head. Again, this is non-polar, so hydrophobic, polar, hydrophilic. Okay, so here is one of these little dudes right here. So now, I'm going to just take one of these. Oops. Okay, so here are a bunch of phospholipids all lined up next to each other. And now, here's the cool thing about this molecule. If we are in an aqueous solution here, which means a water solution, so we have water outside here, these phospholipid heads really like to interact with this water, and these phospholipid tails really like to interact with one another. So they serve as a really good barrier between the water and the phospholipid. But what happens when you have water on both sides? Water over here and then on this side as well. Well, the reason why it's called a phospholipid bilayer is because this actually doubles up. Copy. Ah, so cool. Okay, so you can see what I've done here. I've taken we've created a phospholipid bilayer here. We've got our phosphate heads of one group interacting with the water on one side, and phosphate heads of the other group interacting with the water on the other side, and then the hydrophobic tails interacting with each other on the inside. And this is a very nice position. Cells like to be, or this molecule likes to be like this. But you might ask, well, what about when there's water over here and over here? Well, let's zoom out for a second. I'm going to erase that water here. Now, if you can imagine taking this line of phospholipids and extending it far, 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 far. Okay, until you start to see something like this. Now, this is not a complete cell, but we could take this molecule and totally enclose it. Okay, until we see something just like this. So if we were to zoom in on any point in our cell here, we'd see we've got our phospholipid, one phospholipid right here. Here's our fatty acid tails. Here's our phosphate head. But this creates a complete circle, which is both aqueous on the outside, meaning there's water out here, and also water on the inside. And the phospholipid heads on either end like to interact with the water that's both on the outside or on the inside. And voila! We have created a cell here. This is exactly what your cells are composed of, phospholipids that have aqueous solution on the outside and on the inside. It is a ball of fat. And the really amazing thing about these structures is that if you put phospholipids in water, these cells will form spontaneously because it's actually most energetically favorable for lipids to be in this conformation like this, with the phosphate groups on the outside facing the water and the lipid tails on the interior facing themselves. So add some fats, add some phospholipids to water, and you will get a cell. And I just want to draw your attention again. Remember, this is a phospholipid bilayer, and it's called a bilayer because it's two layers, which we have to see here. There's one layer here and one layer here. Okay, so all our cells are composed of these bilayers, but we actually have a special name for the cell membrane. Or the name for the model that we use to describe it. It's called the fluid mosaic model. I'm going to start a new page here. Okay, so the fluid mosaic model, it's called a fluid model because, as I said before, the structure of the phospholipid here, this kink in here, makes the phospholipid a liquid at room temperature. So things are moving around in your cells. When you look at this cell over time, this phosphate group or this phospholipid right here doesn't stay here. It actually wanders around. It intermingles with all these other phospholipids. So they don't stay in a fixed position over time. They actually move around. So that's where the fluid part of the model comes from. And the mosaic comes from the fact that our cell membranes aren't only composed of phospholipids. They also have many other things that stud the membrane as well. I'm going to enter a picture in here for you. Okay, so here's a picture of the fluid mosaic model. Again, as I said before, it's composed of a lipid bilayer. So you can see here we've got our phospholipid, our head right here, and our two fatty acid tails right here. But also embedded within the membrane are little molecules of cholesterol, which are other lipids that we'll talk about more about over the course of the year. But the amount of cholesterol in the membrane helps determine its fluidity. The more cholesterol in the membrane, the less fluid. So cholesterol gets a really bad rap, but in actuality, it's helping you regulate the fluidity of your membrane. The more cholesterol you put in, the less fluid it will be, and the less you put in, the more solid it will be. So you can imagine that this would be important in an environment where you are changing temperature all the time. What happens when you put butter on the stove? Well, it melts. You wouldn't want your cells to melt when you walked out of the air conditioning. So cholesterol is helping regulate the fluidity of the membrane. Additionally, there are lots of different proteins studded within the membrane, both on the interior and also, it doesn't really show here, but on the exterior, many proteins, and also through the membrane. So these proteins are doing many, many, many different things, and we'll talk about the functions of proteins later on, but proteins are helping things pass through the cell, inside and out, kind of like gates. They only want to let certain things into the cell. Remember, we want to protect our interior, the interior of the cells on the side, from the crazy reactions and daggers that are on the outside. So proteins are only letting certain things pass through. Additionally, we have oligosaccharide. What do you think this is? Oligo, many, saccharide. So another word for a polysaccharide would also be an oligosaccharide. Side chains, these are carbohydrates that are on the side of cells serving as signals, so telling cells what they are. And it's like a flag here saying like, I am a red blood cell, or I am a leukocyte. Any type of cell, they're going to have carbohydrates on the side of them signaling to other cells what they are. And again, all these things are moving around within the membrane itself. The phospholipid phospholipids themselves are moving around. So this one, if you tag this one molecule 5 minutes later, you might see it over here. And same with tagging the oligosaccharide. It's over here right now, but a couple minutes later you might find it over here. And these cholesterols as well, they're diffusing through the membrane. So fluid model, things are moving around. Mosaic, it's studied, you all know what a mosaic is, a series of tiles, right, that make a picture. So we don't just have fats, but we also have proteins and carbs as well, which makes this a mosaic. So it's a fluid mosaic model. Okay, that is it. Yes, ciao.